Welcome back to Translating Proteomics. Today, I'm delighted to introduce you to Fiona Ginty, who is a senior principal scientist in Precision Diagnostics at the GE Healthcare Technology and Innovation Center. She is a leader in the development of spatial proteomics technologies and their application in precision diagnostics and medicine. And among the many other proof points of the importance of her work, Nature Methods named spatial proteomics the method of the year in 2024, highlighting the incredible potential for spatial proteomics technologies to expand our understanding of biological complexity. Additionally, Fiona is a leader on projects within NIH's Human Biomolecular Atlas program, or HuBMAP, which aims to create an open global atlas of the human body at cellular molecular resolution. Fiona, I'm really honored to have you here today, and welcome to Translating Proteomics.
Thank you, Parag, and it's great to see you again. We work a lot together.
Yes, no, it's been such a fun collaboration. And I was thinking back to when we first met. And I went back and I realized it was in February of 2013.
Mm, yes,
So
exactly.
it was a while ago. And so you had come to visit us at Stanford at the Canary Center and shared for the first time I'd ever seen this technology for studying multiplex single cells. And I remember thinking, and I may have even said out loud, wow, that is the coolest thing I've ever seen. And then that led to an amazing partnership and multiple grants together and so much fun. And so I'd love to go back a little bit in time just to understand how you got to where you are and how you first got into biology and your early years studying bone biology at University College Cork. Maybe just share a little bit about the earliest part of your journey.
Sure. Yeah. Thank you for going back down memory lane. It's always fun. So, yeah. I mean, biology has always been of great interest. I think when at school level, in high school, it's very much focused on zoology and the earthworm, and you're not really thinking too far into the future. But it's cool and it's interesting. And so I think, yeah, going to university, College Galway, University of Galway, we call now, I studied microbiology and just became, passionate about bacteria and what bacteria could do. I just thought that was incredibly amazing that you could use bacteria for so many things. And but where I sort of got my interest more in the applied side of biology was in I did a course on nutrition for 1 year and just became fascinated by the idea of biomarkers. And at the time, I remember my professor at the time, Professor Foxwell, he talked about nutrition biomarkers, and I thought, what are they? That just sounds cool. So then I ended up going to Cork and working on in bone biomarkers, and that sort of led me to and did a PhD in nutrition science, and became more appreciative of what biomarkers can do in science, in biology, in clinical translation. So I think biomarkers have sort of been my North Star and along the way. And then from there, went and worked for Nestle for 5 years or 2 years and saw more of the applied side of nutrition, how food products can modify health and,
Well, that's already pretty interesting because I think most people, when they think about Nestle, they think candy bars.
yeah.
But of course, Nestle has just an incredibly deep amount of research going on.
Yeah, it was an incredible research center. It is an incredible research center. And one thing I do remember when I went there for my job interview, there was chocolates there, you could have like, I don't know if they still do that, but that was a very left a big impression on me.
Well,
But
when you're a young scientist, that is a huge draw.
like mega, yeah. So, yeah, I think what was fascinating about the research center, which stuck with me, was it's multidisciplinary. It encompassed, okay, with nutrition, whether it's food science, food technology, as well as the whole sort of field of biomarkers and how you could measure the effects on health. So I think that is something that stuck with me too, is just the team science aspects of how important it is to work with other disciplines.
I'm so curious about how you went from first studying and learning about fundamental aspects of how we detect what's happening in the body and how we alter that with nutritional programs to then building entirely new technologies. And so I'm curious about, was that an explicit mind shift, or that you like, wow, I really wish we had better tools, or how did that transition come about?
Yeah, so I think it was gradual. So I one thing I came to appreciate through nutrition research and still do is how it's not one measurement that determines your health. And it's not one nutrient that determines your health. It's sort of the balance of nutrition, different, for different vitamins, so forth, fat, protein. So it's a sort of a system of systems. And then the measurement tools are so important. So it's sort of like my mindset was already kind of like, okay, all these things have to work together to produce a perfect outcome or near perfect outcome. But I think the thing that's hard when, as a biologist, for me, this is what kind of drove me, I think, into the new technology space, was that you're limited to the tools that you can buy off the shelf. And you always just have more questions, if I could have one more measurement, I could do this, but you're still limited by, what's available. So, but an opportunity came to move to the US and move to GE. And yeah, at the time it was like, when I first started considering GE, I didn't realize what.
Right. Again, when people think about GE, they think light bulbs, but they forget that GE has this whole other aspect to it with MRI scanners and so much wider. And also where you went in GE research, most people are probably not familiar with GE research. So tell us a little bit about what GE research was and its mission, and how it's sort of academic, but also sort of industry and living in the middle.
Yeah, it's a very unique model. I mean, often it's really compared to Bell Labs. It was one of the last industrial labs in the country. It's 1,000 scientists, engineers on site. I joined the biosciences group, which GE had just bought Amersham, and known for the pharma tools, molecular imaging. So we were building a new department, but the fact that there were so many skills that came from other backgrounds that were non-biology actually started to bring together a lot of new ideas. And we were tasked with, our mission was to do for pathology what had been done for radiology, in terms of going from film to digital radiology. And so our mission was to go from slides to digital pathology. Because the problem with pathology, as you know, is, if you've got one piece of tissue on a glass slide, and that was used by the pathologist to make the diagnosis. And then if there needed to be a second diagnosis or referral, then that glass slide had to go be hand delivered, essentially.
Like people were physically mailing around slides.
Exactly. You think about it. I mean, I know it still happens, and there's different, but at the time that was pretty much the only way it was done. And yeah, I mean, and there's so much data in an image. That was the thing I think that was compelling for us. And on the one hand, you sort of take the engineering view. Okay, we're going to scan this slide and we're going to do it really quickly. I mean, we knew what we needed to do for it to be successful. It had to be scanned in a very short amount of time, and then all the QC around that. And there was a big team.
Who was on that team? Were there, because I'm and what, so timeline wise, this is 2009, 2000 or earlier.
It's 2005,
2005.
5 or 6, actually. Yeah. So.
Wow. And so just even the technology to take a picture and digitize it was not what we're accustomed to today.
No, and the team was just like, I mean, it blew my mind, that the none of us were pathology, we were not trained in pathology. There were certainly people on the team who had expertise. And so they were able to, sort of help guide at least the initial questions. But, we worked with pathologists who helped guide us in terms of what they needed, and we watched their workflows and went on site to look how they did things, all the barriers along the whole workflow, how long it took to read a slide. And they're just very simple things that just gave the team the ideas that needed to be implemented. So and I think that sort of quickly evolved from just digitizing the slide to the imaging of the slide of the proteins and the information that were sort of locked within the image. And that sort of was the eureka for me. At first, I was just fascinated. Okay, this is cool. I had never worked in pathology, but the fact that this was, these are, I'm a visual person, so you can see there's information there, but what does it mean?
Can you maybe walk me through a little bit that transition from I'm just trying to make pathology digital to I actually see that there's this space of spatial biology and molecular spatial biology that happened in your head?
Once we figured out the multiplexing part, which is the iterative staining on the same sample, I mean, a lot of that work is going back to 2008. And, we were certainly, just didn't know what to expect in terms of how it would look. The early images were, not great quality. And so once we kind of got...
So it was already 2008 that you were using protein fluorescence, fluorescent imaging on your pathology data.
Yeah, I think we might have even produced the first images in 2007. I mean, we were doing several things at the same time, and some of the team were focused on the chemistry. How do we exploit the fact that these dyes are not stable? And so we had an amazing team. Anup Sood, he came up with the original concept of how to, inactivate the dyes. And so we tried other methods too. We tried light, we tried, different compositions, but the chemical inactivation was the most conducive to doing a whole slide, versus light, it's more focal. And so, those first experiments, we had to prove out how many times could we iterate over, 100 rounds of staining. And I remember another team member, Max Seal, he said, let's just keep going. I'm just going to keep staining and staining and staining. And we were like, okay, are you done yet? Are you done yet? And so, and then another team member, Chris Savinsky, he wanted, again, he had, the early vision, hey, let's get immune cells in there as well. This was, maybe 2009 or 10. And
oh my gosh. So I had no idea that this was happening that early, because I feel like your first multi-omics paper was the 2013 Gerdes paper. Is that?
That's right. So we actually, so Michael Gerdes is another critical team member. He had the vision around combining it with fish and amongst other things. So we had a lot of really great brainstorming during that time. And then we were trying to couple it with the imaging workflow. So, there was always this what if, but we need to do this, but we need to register the images. And so we had, another engineer in the team, Kevin Kenny, who was helping us register all the images. And then there was the autofluorescence. So we knew that was going to be a confounder. So the first data set, I think, probably was about 2008, 2009 that we could actually do some analysis on.
Just to explain for a second, sort of come forward in time, just what the general workflow and approach of cell dive multiomics is, and then we'll go back and dive into a little bit more how we got there. Because I think this concept of iterative staining and dye inactivation, like how does CellDive actually make a multiplex single cell measurement?
So in conventional immunohistochemistry staining, standard what you do is use an antibody that's, say, labeled, it has got a tag on it, some kind of signal that can be detected by light. And the basic principle is you use an antibody, but attached to a fluorescent dye. And then that antibody binds to the protein on the tissue. So it lights up wherever that protein is on the tissue, somewhere in a cell or multiple cells. And so that gives you one photograph or one picture, but then you can get rid of the fluorescence dye, that the tag that's lighting up that cell, and then you can add another antibody with could be the same dye or a different dye. So you're basically taking multiple pictures of the same sample in an iterative workflow, and you're inactivating the dye every time it dyes. And that way you're building up like a montage of pictures of the same sample.
And then because you register them, you're then able to say, I had this marker, there was this much of it in this particular cell at this coordinate, and then marker 2 and marker 3 and marker 4. So you build up this data cube where you've got, some large number, 40, 50, 200 different proteins measured on a per cell basis.
Exactly. And I think that was sort of one of our key things that we got into early, because there had been other multiplexing methods before us that did things a little differently, either with light or had a chemical removal step. But we needed a gentle process, because the sample needed to survive 30, 40 times, where it was going to be stained and restained. So it had to be a gentle process. And then secondly, we wanted to work at a single cell level rather than just have a kind of a lot of data pixel level. That would be hard.
So that was an explicit choice to say we actually want to view this from a cell perspective, not just a pixel perspective.
Exactly. And I think the first thing that drove us as well was we were really thinking from a clinical point of view at the very beginning, meaning, if you take breast cancer, for example, that takes, HER2, estrogen receptor, there's 3 or 4 proteins that are important from a diagnostic perspective. So our view, at least in the beginning, was, well, wouldn't it be cool if we could do all that on the same slide? That's not actually where we ended up, but that was our mindset, that we wanted to measure at a cell level. It wasn't even at the very beginning a single cell level. That came a little later. We were just looking at sort of overall signal within a sample. Was it above a threshold or below a threshold? But then we realized, oh no, we need to actually go to single cell.
So that's a really interesting, because there's a moment in time, and I think my lab was converging around this moment in time, where it felt like as a community we came to appreciate tumor heterogeneity. Yeah. And so I'm wondering if it was that moment where you're like, oh, actually it's not just the total signal. We've got all these different variations from cell to cell that we want to capture.
Yeah, I think that was I think that was sort of an important learning point for us. Once we stopped only thinking about trying to replicate like a current workflow, which at the time was the pathologist's workflow of whether a sample was positive or negative for a given biomarker. We started to work collaboratively with Eli Lilly in about 2010. That was another awesome leap for us, because now we had sort of rudimentary tools developed, but we needed to show them off. We needed people to sort of see, try to understand from a pharma perspective what was important from drug response perspective. And heterogeneity sort of slowly evolved out of that. But once we knew how to do single cell analysis, we were able to start asking those questions because you could see it in the images. It's not like,
Yeah.
it's sort of, you start to look at these stains overlaid with each other. You're like, oh my, there's a cluster of cells there. And there's sort of similar cluster of cells there. And oh, there's immune cells there too. And so you start to think about things in a little different way that we've never kind of thought of before. And then how do you quantify this? It's like that was our next kind of conundrum. How do you convert what you see into a number?
That's right. Because the difference between, hey, percent positive with there's no quantitation. There's just like, is it above threshold or below threshold? And or percent positivity. It's a really straightforward quantification.
Yeah, exactly. And I think that's where we were still kind of thinking, I suppose, bound by thresholds, and that being the only way to understand like the biology. If it's above this, then it means this. If it's below this, it means that. And so we're able to segment every cell in the image. And
Which is a non-trivial problem. Like that is an
yeah,
incredibly hard problem. And for some technologies today, that is still a mostly unsolved problem.
yeah, that was really challenging. We tried to do something fairly straightforward, which was to use a membrane marker. So we had DAPI for the nucleus, we had cytokeratin, and we had the membrane marker. And so we said it, we had sort of the key elements of a cell, if you like. I mean, it wasn't perfect,
Mhm.
but we were able to at least segment the cells. And what we realized in doing so was that you could conserve the spatial coordinate and generate an ID for each cell. But we still didn't know what to do next. We had a scientist on the team who was working with another scientist. This is often how things work in that environment, who had worked in geospatial
Mm.
mapping. Colin was his name. He said, okay, let's talk to Tom because Tom has done this geospatial analysis. I think that might help us. So we explained to Tom what we wanted to do, that we wanted to map the cells and look at their, the neighborhoods or see what they were co-localized with. And Tom said, oh, that's easy. We do that all the time.
We frequently register thousands of images together. No big deal.
Yeah. And so we were like, okay, here's our data set. And then he came back like the next day and showed us, again, very rudimentary co-localization of the different cell types. And we were like, oh wow, that's what we wanted. And so that sort of was our step one. But they were all really long steps, you know.
Yes. And was there a moment there, and when was it that you sort of got that data set that you're like, wow, this this works?
I think one of the eureka moments was we were working with Eli Lilly at the time. They had given us samples that, were drug treated in some way. So, in theory, there was going to be a heterogeneous response. It was, we kind of knew that going in, and the question was whether we could quantify that or not. Taking all the learnings that we had around single cell segmentation, mapping them back to the image, and then I remember another one of our team members, Megan Roth, and she had applied a clustering, k-means clustering method to identify locations, to find the common groupings of cells or cells with common characteristics. And then because we had the spatial location, we could see where they map back to on the sample. And it sort of was amazing. There was sort of, you could see this gradation of, where there was, pockets of either response or cell types. And so we thought, wow, that looks really cool. And then I remember we showed this to Lily and they were, wow, this is the Holy Grail. And we were like, oh, if it's the Holy Grail, well, that's amazing if that's the Holy Grail. And I remember, Chris Vinsky coming in one day and says, hey, we really need to start adding immune cells into our workflow. And
Because you were focused so much on the epithelial cells, probably not even the endothelial cells. You were focused on the cancer cells.
Exactly. And, we're like, okay, let's put them in as well. And then we started getting these beautiful images. Chris was doing this colon cancer study, which ended up being part of the PNAS paper that published in 2013. And we could see, oh, this is incredible. And then Immunoscore had just come out at the time as well from Jerome Galan. And so we're like, oh, this is actually really big. This, we need to understand, we need to have immune cells as part of every study that we do going forward. And so yeah, we were having fun with this, but we also knew we were working towards, we needed to develop something, for GE as well. We
Yeah.
were building a workflow, and I think that was
It needed to evolve from being a, hey, look, we can do it, into something that could be more mature and productized. And
exactly. And I think that was like another transition point for us where we started then to work with Clariant, which GE had bought in, I think, 2011 or 2012. And that was sort of our technology transfer moment that we started to sort of think more practically in terms of how this could work in the clinical context. And that was also very important from a QC perspective, because now we really have to start thinking about workflow. And with the team members, Alex and Bob, and they really guided the team in terms of how do we make something that can be used in a very robust, reproducible context. So that was another big transition.
it sounds like there was a first vision, which was just how do we make pathology digital? And then that evolved into how do we get more information density into pathology? And then you accomplished that, and then had sort of two parallel paths. One was, let's use this and learn things and do science. And then the other was, let's continue to advance and mature the technology as well.
Yeah, exactly. I think, and that was, there was sort of, moments where we were able to do both. And then, we started to work with, Michael Birdie started to work with Vanderbilt, and we got one of our first NIH grants with the team there, and Bob Coffey. And, it was a matter then of being able to prove out the value for the research community as well. So that was one of the first single cell analysis programs that the NIH was running. I think that was maybe around 2014, 2015. But the 2013 paper, that was our watershed moment, in the sense of getting the paper out there. But that took, it's a good 3 years to bring all those pieces together.
Wow. So from beginning to end, it sounds like it was 8 years from the start of the journey, 2005, 2006, to that watershed moment.
Yeah, exactly.
If you think just in the last sort of 3 years, what studies, what projects have you been most excited about?
I think that one of the biggest, most exciting areas that we've worked on in the last, actually, it's been 7 years or 6 to 7 years, is the HuBMAP program, which is the Human Biomolecular Mapping Project. This is a massive effort that spans, we have hundreds of folks on this project. We're mapping the human reference map for healthy organs. Myself, together with UPIT, we're leading the skin atlas and creating spatial map of cell types using spatial proteomics, Cell Dive, Codex, other platforms, similar thing. Map out all the different cell types in our healthy organs, healthy reference organs, and then also combine that with spatial proteomics, spatial transcriptomics, as well as single cell, signal nuclear, RNA-seq. So really multimodal, which I think is where, if you'd asked us 10 years ago or plus, where were we going? I mean, in terms of building these types of spatial maps, we wouldn't have predicted that. So I think that in of itself has been we can chat more about that, but that's been super exciting because it's really starting to show the value of spatial mapping versus dissociated analysis, which is still extremely important from for cataloging and analyzing all the single cells in any given organ. But with the spatial methods, you're really seeing the different layers of cell types, the cell interactions, and then combining that with spatial transcriptomics, then you're really exploding the data.
This sounds like a gargantuan program. Tell me a little bit, how did you become involved in this program?
Well, like any RFA, the call came out. It, I believe, was first approved back in 2016, 2017 timeframe. And then we first applied with our Skin Atlas proposal around that time too, when the RFA came out. And so it's been divided into two phases. So one was about setting up the data infrastructure. All the data has been processed centrally, which is also a huge asset from a community perspective, because it's allowing the data that will be used by the end users. It'll be, we'll have all the provenance and quality control, and it'll be, have a QC stamp on it. We'll also have data that's been analyzed by each of the groups themselves. But this was a massive effort to create that infrastructure, that machine, if you like. We're now in phase 2, which is the production phase, where we're gearing up, ramping up all the kind of with the standardized workflow. So ironed out a lot of the kinks in phase 1. Also, the other interesting thing about this program is that not only are we working in 2D, but we're also working in 3D. And so that's another cool aspect to this, where
so you have multiple sections of the same area so that you're able to dive through different planes.
Exactly. So in our hands, we published our Skin Atlas a couple of years ago. It was in 3D. So we did serial sections, 26 serial sections that were written. Each section was multiplex. So we have 14 or so cell types per serial section. Those are registered, and then a 3D volume is created. So that work was done by Sonia Bose in our team. And then we worked with Indiana to create the tools to interrogate in 3D the distances between, say, endothelial cells and nearest, whatever, your hub cell is. So in our case, we're looking at endothelial cells versus immune cells. That was really at the big, the sort of the proof of concept that you could take this data, register it in 3D, and now start to do some analysis. So that was the basis then for a bunch of tools, including Cell Distance Explorer, which was part of HuBMAP, which is now being used across all other organs in, with different questions about different cell proximity. So I think that's the other important thing about HuBMAP. I'm happy to jump into any aspect of it, but the 3D was important because whilst we're used to 2D and we're familiar with looking at 2D images, organs are 3D, of course. Yes. And so having a 3D reference map is, it's an important step. It's not the end step. There's going to be much more to do in the future, but that was the goal of the program, to try and also do some degree of 3D. So
that sounds incredible. I'm curious, just from when with methods like CellDive, you have to define upfront what markers to look at. And so how did you go about the process, I guess, first of deciding that you wanted to look at skin versus liver or lung, or pick your organ, and then 2, how did you pick what markers to look at even within skin?
Yeah, it's a great question. So for skin, skin is the largest organ in the body. So our collaborators at UPIT, we've worked with UPIT for a long time. Very fortunate to have great collaborators there. And, discussing with them, there was any number of organs we, of course, could have gone after, but we decided skin because, skin disease, inflammation, skin cancer, it's a huge issue that affects everyone at some point in their life. And it's also very accessible. We used samples that in the first few studies that were archived, but then later we were able to get biopsies, which was really nice, from donors who provide us with samples from sun-exposed and chronically sun-exposed, less sun-exposed regions, younger and older. So we're not only doing an atlas, but we're also, at least at a small scale, able to compare anatomically within donors. And so it's sort of, we wanted to bring science into it as well. That was really came about, that whole thinking, we worked with Dr. Lufalo and Dr. Arvarsan Purnamurthy at John and Ho at UPIT, and they just helped sort of form these ideas around how we could approach, skin as an atlas with some interesting questions as well. And then how do we choose the antibodies? Well, the good thing is, when you work with derm pathologists, they know exactly what biomarkers will mark which cell types. They'll, so we wanted, all the epidermis, the dermis, the immune cells, the Langerhans cells. So we documented with them what all the likely markers needed to be, we should include from an atlas perspective. And then separately to this, as a sort of parallel strand to HuBMAP, there's the Human Reference Atlas effort, which is led by Kati Boyner in Indiana, and they're creating this monumental map of all the known cell types and associated biomarkers into one master database, if you like, so that, again, from an external, both from our perspective, could we standardize which markers we were going to select for the most common cell types, map that to what's called the anatomical structures and cell types and biomarkers table that they were documenting with all the other organs.
Yeah, that's really interesting. And in terms of learning so far, I know you're still midstream, but what was the most surprising result for you? And I'm particularly curious in the homogeneity or heterogeneity aspects, particularly in 3 dimensions. It seems like there's potentially some very surprising results that could result from that.
I think one of the really interesting things, which we maybe we're used to seeing sort of the classical pictures of skin in our textbooks, and it's very fixed and it always looks the same. But when you're working with samples from younger, older, sun-exposed, less exposed, then you start to see things like, the skin cell damage and immune cell infiltration. And, in a couple of cases, there was like a minor cyst in the sample, and just so, flood of immune cells. And you come to appreciate how important, of course, skin is and how well designed it is from a vascular perspective and lymphatics and just, the hair follicles, the different cell types, the stem cells, the immune cells. So I think it's sort of, we probably oversimplify skin, but yeah, there's 20 billion T cells in skin. I mean, we caught, maybe 1 million of them.
Wow, that's incredible. That's utterly incredible. So I guess I'd like to maybe change gears a little bit and just talk about, with all of these learnings, what you were doing in HuBMAP was very much basic science, learning the foundations. But with these areas of looking at damage and you're transitioning to thinking about the more clinical side of spatial proteomics. And so I'd love to hear your thoughts on where spatial proteomics is going from a clinical perspective. What are the questions that we're going to be asking with it? What are the places where this might be part of clinical workups in the future?
I think, what's really stayed the course in terms of interrogation, maybe clinical value, translational value, is immuno-oncology, and being able to classify or to image different immune cells and, their exhaustion, their functional status. I think that's still it's still, it's not in the clinic yet. I mean, it will take some time, but it will, to me, that's still probably one of the most promising areas as we try to understand why patients don't respond to immunotherapies. I think it still takes more time. I mean, there's work going on in simulation and modeling to understand, why some tumors are more immunoresponsive than others, what changes that status over time, why is, our older versus younger patients maybe have a different type of immune response. So it's still, I think, there's still a lot of research to do. On the other hand, one of the earliest clinical projects that we were peripherally involved with was with Clariant back in 10 years ago, 12 years ago actually, where the pathologists we worked with there felt lymphoma was really a perfect use case for multiplexing because it can take 20 to 30 different serial sections to determine what subtype of lymphoma. There's over 30 subtypes, I believe, of lymphoma. So, you need to do a lot of sectioning in order to find out, what type of lymphoma that is. I mean, flow cytometry is possible, but you still, you might not have enough sample to do that. So I think, at the time there was a test that was launched for lymphoma, but I feel you might have been a little early, and I think that there's still a need. We, in the later years, it does come back up again that lymphoma is one that still could be, very valuable. And the good thing is that there's a lot more people working in this space now.
So is the thought then that in the future, it may be that we, instead of doing sort of normal single marker pathology, we instead might move to a world where we're doing, full on spatial proteomics and learning much more sophisticated patterns than just how many cells are expressing X marker.
Yeah. I mean, I think there'll be a few steps still to get there because it's the relationship between cytotoxic T cell infiltration and outcomes is still a very powerful predictor. I mean, we see it in all of our studies, even, depending on how many patients you have, but the trend is always there that the higher the cytotoxic T cells, the better your outcome. But then when you start to split it down a little bit more and you start to look at, let's say, the phenotype of those immune cells, you start to see a little bit more gradation there in terms of, the patient, there will still be the patients who always do worse, who have, let's say, cold tumors. But then I think you can split it out further with additional information about those biomarkers, about the functional status of those immune cells. And also, as we get into, the different types of oncology therapies, knowing, what type, the exhaustion status of those, are they functional? Are they able to actually respond? So you could imagine, I don't know if it'll be a 30 marker assay, but it probably will be something closer to maybe 10 at least, where you need to get sort of, and very automated counts. And for sure, there's also, and we showed this actually in one of our papers we published last year on colorectal cancer. The tumor cells themselves, they have to be responsive to the effects of the cytotoxic C cells. And if they're, what we showed in a couple of papers, one was both with the Royal College of Surgeons in Ireland and Queen's University Belfast, we looked at heterogeneity of apoptosis sensitivity and response to chemotherapy. So there's sort of this persistent apoptosis resistant cancer cell type that may be not responding to chemotherapy. And may also, and in another paper, we showed that the proximity of cytotoxic T cells to their neighboring cancer cells, the neighboring cancer cells, you can directly quantify the level of caspase 3 level And
So there's a neighborhood effect. So so me killing this cell actually has an effect on its friends and neighbors as well.
yeah, and I think that whole space is very fascinating because, not work we've done, but others have shown, the sort of the killing time of a cytotoxic T cell, it sort of takes 3 shots, and not just the proximity, even you have to induce
Well, and I think what you're talking about goes even beyond immuno-oncology, because what you're referring to, there's sort of the historical dogma was, hey, I have a kinase inhibitor, or I have a cytotoxic agent, and then it gets to the cancer cell, and the cancer cell is just like, I'm done, I'm out, goodbye, and it dies. Versus there being a necessary interaction with the immune system, even for drugs that are acting on the cancer cell.
Right, exactly. Yeah. The other very interesting thing, I think, is that a lot of these studies that we and others have done, these are adjuvantly treated patients. So the cancer is actually removed, or at least the colorectal cancer, that it's the residual cells that we're making a prediction, or that we're making predictions on a tumor that was
That's not there anymore.
And so, the sort of a very interesting thought that invasion has already occurred, but still we can make some level of prediction. And maybe that's because in the end, maybe the tumors are more uniform from the perspective of, what's left behind.
That's an interesting deep question, because you're wrestling with the reality that the microenvironment in the metastatic niche is very different than the microenvironment in the primary tumor.
Right. Yeah.
And yet you still have this cell endogenous characteristic, and how much is it influenced by its friends and neighbors, and when?
Yeah. And how much training has the immune system already received? There has to have been training that is long lasting. It's a fascinating, I don't know how to solve it. So it's like
well, that's a great segue. So my next question for you was going to be about what can't we do yet? So the technology has advanced incredibly over the last 20 years, but there are still things that we might like to be able to do that we can't do today. So where are the limits of today's technology that you are like, okay, 20 years from now, we should be able to do X, and today it's a problem?
I think speed, throughput, those are, and cost. I mean, they're not the most exciting things to solve, but those are the ones that really...
those are the particular pain points that when you think about the technology achieving ubiquity, those are the big barriers today.
I think, you're competing today against single marker, and you have to prove out the values. And we've seen scenarios where, you're willing to wait as if you're getting good information and you're getting actionable information. Sequencing is, still takes time to do a gene panel. So, it'll be down to the value of the data that you're getting as well. So 20 years from now, I mean, it would be amazing if it was as common to do multiplex imaging as it is to do flow cytometry or sequencing, that it's just built into the workflow. I think we would need to have, a sort of, at least a sort of a 24-hour turnaround. I think things like HuBMAP, HTAN, Human Tumor Atlas Network, and these big efforts will, to some extent, normalize how we think about these new technologies. These are going to be massive amounts of data that are going to be available for further research. And I think that's a tipping point too, because if you look at Cancer Genome Atlas, that's
right. The value of that has been exponentiated because it's been analyzed and reanalyzed and reanalyzed a billion times by so many different researchers and brought in to supplement so many other parallel studies.
Exactly. Yeah. And I think, that's in a sense what it's going to take as well to get us to the ahas. Yeah, I don't think our brains have even got there yet. But I can see, I mean, there's, heterogeneity, types of tumor cells themselves, the different types of immune cells are present, infiltration, location, those are being studied at great depth, I think, across, many publications starting to come out now, as well as sort of like that more basic level of research of understanding what the transitions from early stage cancer to progression, they're having these types of spatial insights will give us other new ideas that we hadn't even thought of.
So on that topic, so my last question for you today, you mentioned Somia earlier, and this, of course, the topic du jour is AI and how this interacts. And so I'm curious to hear your perspectives as we start building these libraries, as we start building these resources, as we start looking not just in 2D, but in 3D. What are you already seeing in terms of AI tools coming to bear? And what do you envision for AI in the future?
Yeah, I mean, Somya goes, he's the right person to talk about this. He's one of our team members and has always been the head of the curve on how we can analyze this data with the latest AI methods. I mean, some of the stuff that we've done in the last few years have included the GAN approaches, generative adversarial network. We're using that to simulate more data beyond what we even have. But I think it's the general idea, when we published that with Vadi and Emory a couple of years ago, looking at H&E images and creating additional patches derived. So you start with n number of samples and, let's say 20 or 100 samples, but then you can start to identify which regions within the sample are associated with aggressive disease and which are, let's say, more innocuous regions. So you're trying to essentially balance your data set by creating more of these aggressive patches. And, one of the problems with these types of analysis, predictive analysis is imbalance. You have a very small amount of aggressive regions, right, and then you have a lot of normal. So I think it's sort of fascinating to think that we can, create more data. You sort of wonder, okay, it's real because it's based off of something that was real, but how far can you go? And that's, I guess, a general question for the field. But
it's a deep question because it was actually quite a surprising result that you could add noise, rotate, do these very simple transforms, and the AI methods would actually learn better.
Yeah.
And as a human, we're like, well, it's the same image. It's just rotated 90 degrees. Why should the AI do better? And yet they did. It was astonishing.
Yeah, no, it is astonishing. And as well, because these technologies are expensive. So can you create more data, without having to spend a lot of money? But, the question is how diverse can you make your data sets and, our rare cell types, they may not be captured. So I think it's sort of got its place depending on what your question is. And then I think also, the deep learning methods for cell analysis and, features within tumors using the H&E, that's another whole other area. But, there's a lot within the H&E image that is untapped as well. So that could also be, a very nice hybrid. But yeah, I think AI can help on all those fronts, also the large language models in trying to just distill down what information, actions, what does it all mean?
That makes a lot of sense. So I'm going to put you on the spot with my last question, which is really, again, a little bit of a forward-looking. If you think about yourself 10 years from now, 15 years from now, and you think about, you're looking back, what have you learned? So you've had all of this time, you've have all of this infrastructure, you've done so many of these big projects. What is that exciting thing that you've learned over the course of this journey?
Probably the immune response is really something very remarkable and not surprising, but how much, I think I hope in the next 10 years that's even more solved than it is today. But just when I look back, over the last 10 years, when it was even maybe more than last 10 years, maybe that's 15 years ago, we did not even analyze the immune cells.
You just ignored them. They were there, they were annoying.
Yeah, just there. They were like, no, they don't need to look at those. And then, but I think it's just profound how that's changed, and just the variability amongst the functions of the different types of immune cells, their interactions, their crosstalks, their metabolism, how that plays into vaccines for cancer vaccines. I think, that's still to pan out, but I hope that's another holy grail.
Anything else that you're excited about as this field develops and evolves?
Yeah, I think the really wonderful, phenomenal thing is just all the next generation of scientists, junior investigators who are now working on this data in totally different ways. We see this in HuBMAP with the junior investigators and their Jumpstart program, and just phenomenal. I mean, they're coming up with amazing ideas around analyzing cells, neighborhoods. You've got people like John Hickey, who's now in Duke, and Chen Zhu in Stanford, and others. There's like a list of folks who they get it, and they're just thinking in a totally different way. And I think that's going to change the field as well. So that's very exciting. I can't wait to see what they do in the next phase.
That's awesome. Well, thank you again. So for our listeners, you've been sitting here chatting with Fiona Ginty, had an incredibly fun conversation learning about the HuBMAP consortium and the role of spatial proteomics in the clinic and where it's going, as well as the future involving AI and the immune system in cancer as well. So thank you so much, Fiona, for joining us today.
Absolutely. Thanks, Parag.
All right. Thanks, everyone, for joining us, and I hope to see you again soon on another episode of Translating Proteomics.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio.